those that aren't here. All right, so the forces of evolution are essentially natural selection, mutation, gene flow, and genetic drift. Also sexual recombination that happens during meiosis that we learned about in chapter 2 and 3. Darwin developed a theory to explain how life on Earth changes, or how to explain how things evolve. His theory was evolution via natural selection. He didn't exactly understand the other forces, but now we're starting to understand the other forces more in depth. But evolution, in a sense, is allele frequencies changing from generation to generation. So we've already learned a little bit about alleles when we did Punnett squares. So we've learned about the various genotypes from Mendelian traits, homozygous dominant, heterozygous, homozygous recessive. And we can use Punnett squares to help determine the likelihood of generation 1 inheriting certain traits. But this lab is going to go more into the what we call the Hardy-Weinberg equation that allows us to calculate the rate of evolutionary change. So evolution is defined. A definition of evolution is a change in gene frequencies in a population over time. So essentially that's what we're going to measure using the Hardy-Weinberg equation. So the rate you see a trait in different generations generation 1 will be different from the rate in generation 2. So if these rates are changing, if you're seeing that the frequencies and the percentages for homozygous dominants versus heterozygotes versus homozygous recessives, if those rates are changing, then you know that evolution is operating on that trait at that time. So essentially what causes these changes? So there are what we call 4 forces of evolution. So natural selection, that's the idea that within a breeding population, within a gene pool, some individuals will possess characteristics that are beneficial for survival, and others will not possess those characteristics. So obviously those individuals that have the beneficial characteristics will be more likely to survive and more likely to reproduce, thus passing along their genes to future generations. And those that do not have those beneficial traits will be more likely to die out before reproducing. Gene flow is essentially just talking about population A interbreeding with population B. So if you imagine that, population A has 100 humans and population B has another 100 humans, if they all start breeding with one another, then that's going to increase genetic diversity. So gene flow is always a good thing, because in the game of evolution, diversity always wins. So genetic drift, we're going to talk about 2 different types of genetic drift, but just for now, know it means a random sampling error, and it generally results in a reduction of genetic variation. Mutations, so mutations are not necessarily always bad. They can be, but mutation is essentially just any change, any random change in the DNA, in the genetic material. So there are some mutations that are actually beneficial. We'll talk about some mutations that give us, they reduce our susceptibility to certain diseases and disorders. There are some mutations that create variation in the gene pool that's necessary for organisms to survive. So mutation isn't always negative. It is sometimes positive, and sometimes it's neutral, meaning it has no effect whatsoever. All right, so the first 3, natural selection, genetic drift, and gene flow, result in the frequency of genes in a population by redistributing. So they result in change by redistributing the existing alleles. So similar to recombination during meiosis. So think about this giant gene pool, and all of these genes are being reshuffled. But with the forces of evolution, there is more intention to it. It's not quite as random, especially with natural selection, because natural selection is selecting those traits that are beneficial, and it's selecting those individuals to survive. Mutation is the only one that introduces brand new information into the gene pool. So again, it's just any random change in the DNA during DNA replication, during protein synthesis, mitosis, meiosis, that's going to create new genetic variation in the gene pool. All right, let's talk first about natural selection. So Both Charles Darwin and Alfred Russel Wallace independently formulated the theory of evolution via natural selection. Wallace actually is one of the reasons that Darwin went ahead and published his results, because he was fearful that Wallace would gain credit for his ideas. They discuss the struggle for existence, extinction of species, adaptation, and variation. So they realize that within a breeding population, more organisms are produced and can possibly survive. So that's where natural selection comes in. Once a species has exceeded the carrying capacity, that's when natural selection really comes into play. Darwin looked at the work of Malthus, who said that resources are limited, but population growth is not. So essentially, a population size will continue to grow even though resources within that given habitat are limited. So exceeding the carrying capacity means when the population size has become so large that they can no longer fully feed and support all the organisms within it. I mean, I'm sure you could certainly argue that human beings have exceeded the carrying capacity on the planet. We have over 7.5 billion people on the planet, and there's certainly a lot of people that don't have enough resources to survive. So Darwin realized that selection acts upon an individual, and this led him to formulate the principle of natural selection. All right, so here are the steps of natural selection. So it's important to keep in mind, like I mentioned before, that more individuals in a population are produced than can possibly survive. So that's when we start exceeding the carrying capacity. Once they reach that point of crisis or that carrying capacity where individuals are starting to starve. So there is variation, and some individuals are better adapted to their environment than others. So just envision this very varied gene pool with individuals that have a variety of different traits. So some individuals will possess the traits that give them an advantage. Let's say we're talking about the rock pocket mouse, for example. So, you know, the rock pocket mouse has two different colors, a sandy brownish color and a dark gray color. So at the point in time when their substrate, when their environment was completely filled with the sandy desert, obviously at that point in time, the rock pocket mouse that had the advantage was the sandy color, because they were able to avoid being eaten by aerial predators like hawks and eagles. But when the environment shifted, when the rock pocket mouse now lived on an environment where it was almost blackish in color because there was a volcanic eruption, and then once that lava dried, the substrate was blackish in color. Now all of a sudden the selective pressure shifted, and now all of a sudden it's the dark colored mouse that has the advantage. So individuals will compete for limited resources. So they compete for food, for access to mates, for access to territory. So it's generally those that have the most advantageous traits will be the ones that are the most successful at that competition. So the better adapted ones will survive and reproduce and essentially pass their genes along to future generations. So what happens over time is beneficial traits end up increasing in the population and detrimental traits decrease. So what this results in is gene frequencies changing over time, which, as you remember, that's the definition of evolution. Evolution is essentially the change or the fluctuation in gene frequencies over time. And also, if populations become isolated, this may result in enough genetic differences to create a new species. So if a certain subset of a population becomes isolated on an island, let's say the lemurs, for example, you know, the lemurs originated on the mainland of Africa, and then some of them were able to migrate to Madagascar, and they went through a process called adaptive radiation. So they adapted to fill specific roles, specific what we call niches within their environment that were very different roles than they played on the mainland of Africa. So over time, these lemurs, they adaptively radiated to fill these very specific roles, to eat specific types of food. Some of them were active during the day, some were active at night. So essentially what happened is over time, we have all of these different species of lemur. We have dozens of different species of lemur living on Madagascar that are completely genetically separate from the lemurs that lived on the mainland of Africa. So what happens if you have enough geographic and genetic isolation, that founder population eventually becomes separate from the parent. So it means they can no longer interbreed with the parent population and produce viable fertile offspring. All right, so another example is, of course, Darwin's finches, very famous example. So Darwin noticed that a lot of his finches that he was studying had, they were similar, they had similarities, but they also had some pretty stark differences. Like, for example, you've got this finch here with the really thick beak, and then you've got another finch over here with the really tiny, skinny beak. So he wanted to understand why this happens. So what he realized is that all these finches are eating different types of food. The finches with the really thick, stout beak, those finches are eating harder food items like seeds. And then those finches with the really long, skinny beaks, those finches are eating softer food items like fruits or nectar from flowers. But when you really think about it, that's beneficial to all the finches and the entire habitat. Because if you can envision, if all the finches were trying to eat the exact same food type, it would be much more likely that they would come into competition with one another. So it's actually beneficial that all of these finches have adaptively radiated to fill various niches, various roles in their environment. So if to give you a simple example, if you imagine that there's 100 of us in a room and we've got 80 different candy bars, and if most of us like Snickers, but we've only got 10 Snicker bars and 10 Butterfingers and 10 Twix bars and 10 Milky Ways, you guys get the point. But if 80 of us like Snickers, but there's only 10 Snicker bars, then obviously we're going to compete with one another to get those 10 Snicker bars. But if 10 of us like Snickers and 10 of us like Butterfingers and 10 of us like Milky Ways and 10 of us like Twix, then we actually don't come into any competition with each other, because we're all able to get the food that we want without having to compete. So, I mean, that's just obviously a very simple example, but if you think of it that way, it's kind of the same idea as the finches, same idea as the lemurs. They're adapted to eating very specific foods and living in specific environments, which actually is key for all of them. That variation is key. All right, so remember, natural selection acts on individuals and evolution acts on populations over generations. So generally we don't see evolutionary change. We can only measure it if we're able to measure generation to generation. So that's why we use the Hardy-Weinberg equation, and you'll see why here in a moment. So each individual has differential reproductive success, and this results in a change in gene frequencies as well. So some individuals will be better adapted to reproducing than others. So obviously those that are adapted to reproduction will end up passing their genes along to future generations. So the fitness, so that's not necessarily talking about how good you are at going to the gym. That term fitness is talking about both your individual success. So fitness not meaning how well you can work out, just meaning how adaptable you are to your environment. That's your fitness, that's your individual fitness. And there's also your reproductive fitness as well. So the fitness, essentially reproductive success of any variation, will change as the environment changes. So like I mentioned with the rock pocket mouse, just because one color is beneficial for survival in that environment at one point in time doesn't mean that 10 years from that time it'll still be the beneficial trait, because the environment is constantly changing, constantly fluctuating. So it's important to remember that just because a trait is beneficial now doesn't mean it'll be beneficial a year from now or 5 years from now or 10 years from now, so on and so forth. All right. So adaptive radiation, that's the term that I used when I was talking about the lemurs and also with the finches. So remember, it's that idea that once this subset of the population becomes isolated from the parent population and a new environment, they're going to adaptively radiate to fill these new environmental roles or environmental niches. So it's when many new species evolve out of one common ancestral species. So if you envision the tree example here, so let's just say down here we've got the bottom of the tree. So we've got the bottom of the tree down here, that's representing the common ancestor, and then adaptive radiation is all of these branches. So if you envision the bottom here, that's the common ancestor. And then all of these branches are representing speciation events. These are all speciations. So these finches, obviously they have similarities, but they also have differences. They have beaks that are different sizes, they have different colorations, they have different song tunes that they, different pitch to their song. So again, that's beneficial to them all because they're not all trying to eat the same thing. Some of them eat vegetation, some of them eat insects, some of them eat seeds, some of them eat nectars from flowers. They're eating different things, and that's meaning that they're not coming into direct competition with one another. All right, do you guys have any questions about natural selection before I move on to mutation? I'll take the silence as a good thing. Yeah? All right, so mutation. So a mutation is any inheritable change in the genetic material. So remember, it's not always bad. It just means it's a random fluctuation in the gene pool. So mutations can occur during DNA replication, during protein synthesis. We'll talk about the different types of mutation. So think of mutation. Mutations create the variation within the population, and then natural selection acts upon that variation. So natural selection together with mutation can change gene frequency more rapidly. Mutations are completely random. I mean, there are fields of study like epigenetics that are looking at, essentially looking at the possibility that the environment can influence mutation rates, but that certainly hasn't been confirmed. So what we know as of right now is that mutation is completely random. So these can be positive. By positive, it means that it provides a benefit to the organism. They could be negative, meaning that it causes a detrimental effect, or they could be neutral, meaning it doesn't provide any sort of positive or negative effect. And mutations are important to evolution because they are the only source of brand new genetic information in a gene pool. And remember, you're probably going to get sick of me saying this, but in the game of evolution, variation wins. So in that sense, mutation is certainly a very necessary and beneficial aspect of evolution. All right, let's talk about the types. So there are several different types of mutations. A point mutation is usually the least severe. So it changes one base, so one adenine, one thymine, one cytosine, one guanine, out of 3.1 billion in the genome. So most are silent mutations. So even though one adenine might change to a cytosine, for example, it may still code for the correct amino acid. So if it's still coding for the correct amino acid, there's no effect on the organism. So a point mutation can go unnoticed, or if on a specific codon, can lead to a large outcome. So an example of a point mutation on chromosome 15, there's a point mutation that creates a allele that codes for sickle cell anemia. So sickle cell anemia is a disorder that affects your red blood cells. So this, if you get this mutation in the homozygous recessive condition, so if you inherit that mutation from both your mom and your dad, if you're homozygous recessive, so that's the SS genotype for sickle cell, then you do in fact have sickle cell anemia, which is a deadly disorder without medical intervention. Essentially what happens is your red blood cells become sickle shaped, and they clot and they cause blood clotting, and it means that your red blood cells are unable to carry oxygen to your essential organs, which obviously is a very bad thing. So those that suffer from sickle cell anemia, especially in low oxygen environments, they often have organ failure. And it is a disorder that is very serious and can result in death without medical intervention. So of course, those of us that study evolution makes us wonder, why did such a terrible, deadly disease develop, and why did it not get removed from the gene pool if it was so detrimental? Well, the reason is if you inherit it in the heterozygous form, so if you inherit a mutated allele from one parent and a normal gene for hemoglobin from the other, if you're the AS genotype, then you're actually resistant to malaria. So what researchers found, is that the sickle cell gene is actually really common if you live or if your ancestors originated from a region where malaria is endemic, constantly present. So that AS genotype provides you a huge benefit, because especially in regions where where medical support is limited, malaria can also be extremely deadly. So malaria is carried by mosquitoes, And if you get bitten by a mosquito that's carrying malaria, the malarial parasite will finish its cycle within your red blood cells. But if you have the AS genotype, you have a slightly lower oxygen content in your red blood cells, not enough to affect you, but enough to make it so the malarial parasite can't finish its life cycle. So if you're this AS genotype, you have a huge benefit because you don't have sickle cell anemia and you're resistant to malaria. So they found that any region where malaria is endemic, this AS genotype is very common because it provides an evolutionary benefit. All right, so missense point mutation. So a one-letter base is wrong, so it misspells a codon. So remember thinking back to protein synthesis from chapter 2 and 3. Protein synthesis, you've got this, the DNA template codes for the messenger RNA strand. And then they've got a picture of it here. So each messenger RNA strand, each set of 3 is a codon. So for example, CAT is coding for histidine. We got another histidine here, histidine. GGC is coding for glycine. CAT is coding for histidine, histidine, histidine. So the error here, the missense mutation is right here. So you've got this codon, the GGC, which should code for glycine. So that G is randomly changing to an A. So all of a sudden, a GAC is now coding for aspirin. So this missense mutation is essentially meaning that the wrong amino acid is being coded for. All right. So a nonsense point mutation, so it's a premature stop codon, which shortens the length of a protein. So there is a codon, there's codons that code for start, which means the start of that coding protein. And then there's also a stop codon as well, which in this example is TGA. So some examples of that, thalassemia, so CAG, so the C randomly changes to a U, which is, or the CAG, the C randomly changes to a U, which then codes it to stop. So it's similar to the sickle cell example where blood cells are destroyed, leading to anemia, and also McArdle's disease. So the CJA changes to a UJA, which is a stop codon, which results in an inability to produce the enzyme that breaks down glycogen, which is sugar, and it results in extreme muscle pain. All right, so a deletion, just like it sounds, would be a random amino acid or a codon deleted from the code. So it's removing amino acid codon, resulting in a misfolded protein. So an insertion is just the opposite. So just like it sounds, it's added or repeated bases. So adding a base changes all amino acids that follow. So same with a deletion. If you're deleting an amino acid codon, or if you're adding an amino acid codon, it actually affects the entire sequence from there on out. All right, so a common cause is failure of chromosomes to separate properly during cell division. So separation of chromosomes during anaphase is called disjunction. So remember that, remember our phases of mitosis. We have interphase, prophase, metaphase, anaphase, telophase. So during anaphase, that's when the spindle apparatus rips apart the chromosomes at their centromere. So the chromosomes are being ripped apart, resulting in what we call disjunction. So failure of the chromosomes to separate properly is called a non-disjunction. So if a non-disjunction occurs, all of the cells will end up with the wrong number of chromosomes. All right, so some examples of this. So the standard condition, what we want is a disomy. So remember our karyotype, we want a disomy. We want 22 pairs of autosomes and then 1 pair of sex chromosomes. So that's a disomy, that's the standard condition. If you break that word down, di means 2, somy means chromosome. So if we have a mutation where 1 pair is missing, for example, Turner syndrome results when there's 1 X chromosome but not a second. So a monosomy is when there's a pair missing. And then if there's an extra chromosome, we call it a trisomy. So an example of that is trisomy 21 or Down syndrome. So you see here in this karyotype, everything looks normal all the way up until you get to the 21st autosome. So instead of having 2 pairs of 21, you've got 3. So there's other trisomies. There's trisomy 11, which is Pateau syndrome, I believe. Trisomy 18, which is Edwards. So these trisomies, they generally have pretty severe consequences for the individual. I mean, many individuals with trisomies can still live long, happy lives. But a lot of the trisomies that occur on the larger chromosomes, remember, the larger the chromosome, the more genetic material they carry. So a trisomy that occurs on chromosome 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 are generally too severe for survival. So sometimes what happens if there's a trisomy or a monosomy on one of those chromosomes, it'll result in a spontaneous miscarriage. So essentially it's the body recognizing that that mutation is too severe for survival. And, you know, not to get political with you guys, but that's what happens too when you're talking about these debates about female reproduction and reproductive rights. If they take away those rights from doctors and women, when there's those severe chromosomal aberrations that happen, there's no options left. Like sometimes these aberrations are so severe that neither mother or child would survive. So sometimes a medical professional steps in and actually recommends that the pregnancy is terminated. But of course, obviously that's up to the individual, but you know, just to give you guys a little bit of background on that. How you guys doing? Do you guys have questions? Do you have any questions at all? Can you guys just give me an okay so I know you're still there?
Getting it pretty good.
Okay, good. All right. Okay, good. All right, so this one here is moving on to human chromosomes. So as we've learned in chapter 2 and 3, humans have 46 chromosomes in the nucleus of every somatic cell. However, all other apes, so chimpanzees, gorillas, bonobos, orangutans, are going to have 48 chromosomes in the nucleus of every somatic cell. So let's look at why. All right, so what happened is called a balanced translocation fusion. So 2 chromosomes went to the wrong location and fused. So 2 ape chromosomes turned into our human chromosome number 2. So there is actually a YouTube clip here. I'm not going to show it right now just because on Zoom it's not, the visual and audio quality is not as good as if you're just watching it on your own. But this is a really interesting little clip. So if you guys want to click on it from the PowerPoint, or I'll add it to the module as well. So it may also be due to some level of inbreeding. So it's a very, you know, random mutation that might have really, you know, caused or influenced that split. So like I said in previous lectures, you know, we didn't evolve from chimpanzees, but we do share a common ancestry with them. So that could have been what that translocation fusion could have been what caused that split to occur, what caused the hominin line to branch off from the chimpanzee line, essentially. All right, so it's happening again. We do see some examples. There was a man in China whose parents were very close relatives, and they both had a 14 to 15 balanced translocation. And this man ended up perfectly fine, had no detrimental, like lived a perfectly happy, healthy life, but only had 44 chromosomes. So there's also some recorded cases in both Finland and Spain. So some interesting stuff going on. It's very interesting. All right, do you guys have questions about mutations before I move on? Okay, let's talk about genetic drift. So genetic drift, there's two different types of genetic drift. So the first type is going to be called the founder effect, and the second type is going to be called the bottleneck effect. So essentially one generation's frequencies may not accurately represent the frequency of its previous generation's genes or the parent population. So there may be an overrepresentation or underrepresentation of traits due to small sample size. So let's talk first about the founder effects. I feel like it's the easiest to understand. So I want you to again envision the lemur example. So let's say we've got, let's say 200,000 lemurs on the mainland of Africa. And let's say that, let's say 4,000 of them successfully migrate from the mainland of Africa to Madagascar. So by chance, I mean, just envision it as essentially taking a handful. We're going to essentially take a handful of lemurs and place them on Madagascar. So by chance, this handful of lemurs, this 4,000 lemurs, does not have the same genetic variation or variability as the parent population. So what happens once they become isolated? So the founder effect is talking about, they're the founding population. They're founding that territory in Madagascar. So they're going through adaptive radiation. They're adapting to the environmental circumstances on Madagascar. They're adapting to eat specific types of food and to avoid certain types of predators, to live in different territories. Some of them are active at night, some of them during the day, which is again beneficial to everyone. But they became reproductively isolated for long enough that they could no longer breed with the parent population. The parent population is the population back on the mainland. But what can happen if there's too small of a sample size, if let's say only 100 lemurs made it over to Madagascar, then it's very possible that 100 lemurs would not have enough genetic diversity to survive. Because, again, in the game of evolution, diversity wins. So there has to be a large enough founding population for there to be enough variation for survival, which in the case of the lemurs, thankfully there was. The lemurs no longer exist on the mainland because, there's a lot of different predators that wipe them out on the mainland, but lemurs do still exist in natural habitats in Madagascar. All right, so second type of genetic drift is called the bottleneck effect. So I think there's a picture on the next page. It'll help us understand the bottleneck. Okay, so the bottleneck effect, so again, it's a sampling error. So we've got here the parent population. So this first bottle is representing the parent population or the original population. And then all of these different colors are representing different alleles, different genes. So we've got a pretty even mixture of red, blue, and green, right? So then in the next generation, there's some sort of random sampling error. There's some sort of random event that means there's only a few survivors. So if you can see here, most of the surviving individuals are those that carry that green trait or green allele. There's a few with the blue, but only one of the red. So for whatever reason, whatever event happened, whether it was an environmental catastrophe or destruction of habitat or poaching, there was some sort of random event that meant that only a few survived. And then what happens generation after generation, of course, the survivors continue to interbreed with each other. And then your end result, you've got the next population that is predominantly those that have this green allele. And there's only a few of the blue and only one of the red left. So population bottlenecking happens a lot when there's some sort of, it's happened a lot recently, when there's some sort of drastic event, habitat destruction, environmental catastrophe that wipes out a good portion of a population. So let's say we're talking about mountain gorillas, for example. Let's say that there were 500,000 mountain gorillas 30 years ago living in Rwanda and Uganda. And then there was habitat destruction, poaching, the pet trade, the zoo trade that wiped out a significant portion. And today there's only about 700 mountain gorillas left. So that's a population bottleneck. Of course, we all hope that they do have enough genetic diversity, but it's very possible that the extinction of the mountain gorillas is inevitable because their genetic diversity has been reduced so drastically. Of course, I hope that's not the case. I hope they're around for many generations to come, but that is the scary thing there, is that when population sizes get reduced so drastically, it's very possible that it's no longer possible for that group to survive. Because again, in the game of evolution, diversity is what wins. We need genetic diversity in order for that population to adapt and survive. All right, do you guys have questions about either the founder effect or the bottleneck effect? No? Okay. All right. Okay, so gene flow. Gene flow, honestly, is probably the easiest one to understand. It's just essentially generation 1 interbreeding with generation, or with population B, A and B breeding together. So just envision you've got population A that has, let's say, 500 individuals, and population B that has another 500 individuals. Gene flow is just saying that population A and population B are interbreeding, their genes are flowing. It's also sometimes called migration of genes. So in evolution, this is a good thing. We want populations to breed together. So it's two previously separate groups that are now coming together, mating, merging gene pools. If we talk about a population being made up of genes rather than individuals, we're referring to the entire gene pool. So that's all the genes that exist for all those individuals in that population. So if the 500 individuals from population A combined with the 500 individuals from population B, we now have a much larger gene pool. All right, so also too with humans, gene flow is definitely something that happens a lot more than it did in the past because of modern ways of transportation like planes, trains, automobiles. We can definitely interbreed with individuals that don't live in our hometown. We're not restricted to just those that we can, you know, easily get to. We can breed with humans that live on the other side of the continent. We can breed with humans that live in other states, other countries, et cetera, et cetera. All right. So the sickle cell example, which we've already talked about a little bit, but there's some good pictures here. So if a person is homozygous recessive, meaning they inherit, so remember this is a point mutation on the 15th chromosome in humans. If a human inherits that sickle cell mutation for both mom and dad, if they're the SS genotype, it means that their red blood cells will sickle. So that the red blood cells, instead of being nice and healthy like this is a normal red blood cell, nice and circular. A sickled red blood cell has a sickle shape. And what happens is it results in blockage and blood clotting, and it prevents oxygenated blood from reaching all the vital organs. So those that have sickle cell end up generally dying before reproductive years unless there's medical intervention. But we see really high levels in areas like sub-Saharan Africa, India, southern parts of Europe, like Italy. It's not just Africa, that's kind of a misconception. We see this trait really anywhere where malaria is endemic, because we learned that the AS genotype has an advantage. It's a balanced polymorphism. So what we mean by that, the middle, the heterozygous has the advantage. The heterozygous form is selected for because it prevents and makes individuals less likely to get malaria. So if you inherit on your 15th chromosome at the location, at the locus that codes for hemoglobin, if you have completely normal blood, you're the AA genotype, which is good news in the sense that you won't have sickle cell anemia, but it's bad news in the sense that you have a high chance of dying from malaria. If you inherit the mutated allele on the 15th chromosome at the locus that codes for hemoglobin, if you're the SS genotype, then you have sickle blood, which would definitely be detrimental to your survival, because that sickling generally, especially if you're in a region that does not have high quality medical care, generally leads to death before reproduction. If you have the AS genotype, you're the heterozygous form, you're protected from malaria, and you have enough red blood cells to get oxygen. So like I mentioned before, what happens in the AS genotype, you do have a slightly lower level of oxygen in your red blood cells, but not low enough to affect you, the human, negatively. But what it does do is it's a low enough level of oxygen that it prevents the malarial parasite carried by mosquitoes. It prevents that parasite from completing its life cycle. Okay, so if you're the AS genotype, that's what you want to be. That's, if you could pick, you know, if you live in a region where malaria is endemic, if you could pick, you would want to be the AS genotype, because it prevents you from getting malaria, and you also don't have sickle cell. All right, so this disease is an example of a mutation. Remember, it's a point mutation on the 15th chromosome that causes the wrong codon to be coded for. It's also an example of natural selection, because in Africa and other regions where it's endemic, where it's present, where malaria is present, it is selected for in the heterozygous form to prevent malaria. It can also be an example of genetic drift, because in America, this is seen in higher frequencies in African Americans because of the slave trade. So they were brought to a new location. So that's essentially an example of the founder effect. So, you know, just imagine, you know, a certain subset of a population was, you know, brought from their main homeland where it was beneficial for them to have the sickle cell trait because it prevented malaria, or randomly transported to another continent. So, you know, and if you're living in a region where malaria is not endemic, it's not necessarily beneficial to be the AS genotype, because if you envision your Punnett squares, if an AS mom breeds with an AS dad, there's a 25% chance that they will produce a child with sickle cell anemia. So obviously that's detrimental to that population. All right, let's talk a little bit about Hardy-Weinberg. And I'm actually, what I'm going to do is I'm going to bring up a different PowerPoint for this. because I've got one that's easier to kind of explain over Zoom. There we go. Okay. All right, so one thing too, guys, go ahead and if you've got something nearby, if you've got a sheet of paper and something to write with and a basic calculator, we'll actually do one of these together just to make sure you understand. So go ahead, I'll give you guys a second if you want to grab just a blank sheet of paper and a pencil and a calculator. Just a basic calculator is fine. You guys have any questions about anything? Okay, so Hardy-Weinberg Law is essentially a method for studying genetic change in populations. So you're studying evolution. So remember, evolution is studying the rate of gene frequency changes from generation to generation. So if no changes is occurring within a population, gene frequencies at a locus, remember a locus is the location on the chromosome, will remain the same. So let's just say, you know, as a random example, we're talking about earlobe form. So if there's no evolutionary forces that are selecting for a certain earlobe form, then we can expect gene frequencies to remain relatively constant over time. If change is occurring, if there are evolutionary forces that are acting on a certain trait, we know that evolution is occurring within that population. So Hardy-Weinberg equilibrium assumes that there's random mating, that there's an infinitely large population size, that there's no gene flow, no genetic drift, no natural selection, and no mutation. So for a population to be in equilibrium, it just means that there's no evolution, there's no evolutionary forces acting on that trait at that time. That's the null hypothesis. All right, so the equation itself, and I'm going to say this a couple times, not to be annoying, but just this is really important to kind of cement into your head. So the equation itself, p squared plus 2pq plus q squared equals 1. So one more time, the equation itself, p squared plus 2pq plus q squared equals 1. So p is going to represent the frequency of the dominant allele within that gene pool. So p and q, that's talking about the entire gene pool. So p is representing the frequency of the dominant allele in that gene pool, and q is representing the frequency of the recessive allele in that gene pool. So if it's talking about P and Q, you're talking about the entire population, the entire gene pool. All right, and then P squared is referring to the homozygous dominant individuals in that gene pool. So one more time, P squared is referring to the homozygous dominant individuals in that gene pool. 2pq is referring to the heterozygous individuals in that gene pool. And Q squared is referring to the homozygous recessive individuals in that gene pool. Okay, so easiest way to kind of explain this is just to do an example. So this is where if you've got a blank sheet of paper, go ahead and pull it out. Blank sheet of paper, something to write with. So with the Hardy-Weinberg equation, you're always going to start with the number of homozygous recessive individuals in the population. And the reason for that is that's who you know their genotype for certain by looking at their phenotype. Let's say we're talking about earlobes, for example. So if you have attached earlobes, that's the homozygous recessive genotype. So let's say within a given population, let's say we've got 1,000 people in this gene pool. Let's say that 200 of them have the homozygous recessive genotype. 200 of them are unable, or they have attached earlobes. So 200 have attached earlobes. And then we're going to divide that by the total number of individuals in the population. So it's 200 divided by 1,000. So everybody take a second, go ahead and punch that into your calculator. 200 divided by 1,000 gives us 0.20. So careful, this is the value of q squared. This is not the value of Q yet. So this is step one. We take the number of homozygous recessive individuals, divide it by the total in the population. It gives us this number, the 0.20. That's the value of Q squared. But we want the value of just Q. So step two, to find the value of just Q, we now need to take the square root of 0.20. So the square root button, that's the one that looks like a check sign with a line on the top of it. So we take, here, I'll go ahead and pull out a calculator with you guys just so you can see it. So we take out our calculator. So we're in our calculator 0.20, and then we're going to hit this button here, the square root button. Okay, so we're always going to round when we're doing the Hardy-Weinberg equation. We're always going to round to 2 decimal places. So to round to 2 decimal places, you look at that third number. So you start at the decimal place and you count out 3. If that number is 5 or above, you're going to round up. So our value of Q equals 0.45. Okay, value of Q is 0.45. You guys with me thus far?
Yep.
Okay, so we got our value of Q. And then this is just a mathematical element that we know, like since we know that P plus Q equals 1, to find the value of P, all we need to do is invert it. So we just take 1 minus the value of Q, which is 0.45. And that's going to give us the value of P. So 1 minus 0.45 is 0.55. So now we essentially know P equals 0.55. And we know that Q equals 0.45. Okay. All right. So that's essentially, if you can do that, the rest of it's a piece of cake, as long as you've memorized the Hardy-Weinberg equation and as long as you've memorized what each part of it represents. Okay. So one more time, just for good measure, our equation itself is P squared plus 2PQ plus Q squared equals 1. Okay, so when I say plug it in, when I say the words plug it in, plug in the values of P and Q into the Hardy-Weinberg equation, all I mean is anytime you see a P, you're going to plug in the value of P. So P squared is essentially P multiplied by P. So we're going to plug in 0.55, and we're going to multiply it by itself, 0.55. And then for the 2PQ portion, so we're carrying it down here. We've got the 2. And we're going to multiply it by the value of P, which is 0.55, and then multiply by the value of Q, which is 0.45. And then for the Q squared portion, so our last portion of the equation, Q squared is essentially Q multiplied by Q, so 0.45 multiplied by 0.45. Okay, and then next step is next part is we're simply going to use our calculator and do the math. So I'm going to do it on the screen here with you guys just for good measure so you can see what I'm doing. So the first portion of our equation is P squared. So we're taking 0.55 multiplied by 0.55, which gives us this number here. So we look at that third number. So because that third number is 4 and below, we're going to leave it the same. So 0.30 is the frequency for the homozygous dominant genotype in that gene pool, that breeding population, okay? So if we're talking about earlobes, we're using the letter E, so 0.30 would be big E, big E. And if you want to turn that number, this number here, this is a frequency. If you want to turn a frequency into a percentage, piece of cake. All you have to do is move that decimal 2 places. So move that decimal 2 places, we get 30%. So we know that 30% of our breeding population is homozygous dominant at the locus that determines earlobe form. Remember, the locus is just a location on the chromosome. It's where the gene is located. All right, and then middle part of our equation. So that's the 2PQ portion that's going to be representing the heterozygous form. So we take 2, multiply it by 0.55, multiply it by 0.45. So 2 times 0.55 times 0.45. We'll do it here in our calculator. 2 times 0.55 times 0.45. Oops. All right, so then we're rounding to 2, so we look at that third number, and because it's a 5 and above, we round up. So this becomes 0.50. So remember that number, that frequency, that's representing, so you follow it all the way down, the 2PQ, the 0.5 is representing the 2PQ portion of the population, which is the heterozygous genotype. So in this case, we're talking earlobes, so one big E, one little e. So we know, we move our decimal 2 places. We know that 50% of our population is heterozygous at the location, the locus that codes for earlobe form. So we've got 30% homozygous dominant, 50% heterozygous, and then the last portion of the population is our homozygous recessives. So we take the value of Q, which is 0.45, multiplied by the value of Q, which is 0.45. And it's going to give us, we look at our third number, it's below 5, it's 4 and below. So we're going to keep it the same. So this remains 0.20. So that's a frequency there. To turn that frequency into a percentage, we move our decimal 2 places, we get 20%. Okay, and the nice thing about the Hardy-Weinberg equation as well, we can use this, we can add it together to make sure we get back to 1. So we take 0.3 plus 0.5 plus 0.2, we get back to 1, which is representing 100% or the entire gene pool. Or we can also do 30% plus 50% plus 20%. Oh, this calculator. Sorry, 30% plus 50% plus 20% gets us all the way back to 100%. Okay? All right, how are you guys? Do you guys have questions about the Hardy-Weinberg equation?
Are these instructions for the worksheet, are those also in the modules, part of the lecture? Because I couldn't find them.
Oh, so if you're in my lecture class, this is actually check-in assignment number 2. Sorry, I added another check-in assignment. So if you're in my lecture class, what we actually went through is practice problem number 1, check-in assignment number 2. Okay, and yeah, this PowerPoint I actually pulled, if you saw, I pulled it up directly from our Canvas page. So I'm not sure why you weren't able to access it, but let me know. I mean, let me know if you're having trouble. But
okay,
doing the Hardy-Weinberg equation. Okay, yeah, doing the Hardy-Weinberg equation is going to be important for both lecture and lab. You'll be tested on it in both classes. So I mean, you know, and if you're in both, even better news. You can kill 2 birds with 1 stone. If you learn it in this class, you'll know it for the other one. But I'm sure that's the case for most of you, even some of you that may have different lecture instructors. You will probably need to know the Hardy-Weinberg for both your lecture class and your lab class. Good question, though. Other questions? No? Okay. Do you guys want another example, or do you feel good about this?
I feel pretty good.
Okay. Is there anybody out there that would like a second example of how to do the Hardy-Weinberg?
This will be available for me to look at later again on the recording, correct?
It will, but guys, I've been having some issues with Zoom lately. Like I recorded a Zoom on Monday that still hasn't been transcribed, so I can't guarantee when. I mean, yes, as soon as it's available to me, I will post it for you guys, but it may take up to a week. So, I mean, my advice to you guys, I mean, especially, I mean, I assume it's because literally every teacher in the country is, in the world really, is using Zoom. So it's just taking them a lot longer to transcribe. So, I mean, my advice is obviously to be here for the Zoom meetings and to ask if you want another example. You know, I'm happy to sit here with you guys and do another example. So, you know, my advice is if this is a little fuzzy, let's do another example now, because I can't guarantee when this Zoom recording will be available. It could be a week. Would you guys like another example, or are you feeling okay?
I don't mind another example.
Okay. All right. So I mean, I guess, I mean, if you're one of those people that feels good, you don't need another example. I mean, you're free to log out now if you'd like, and then if you'd like a second example, go ahead and hang in there. And I'm going to pull up a blank document so I can give you guys another example. All right. I need Microsoft Word. Okay. All right, so let's say we've got a population. Oh, I gotta go back to share screen. Okay, so let's just say we have a population of 100 zebras. And let's say out of the 100 zebras, we have, oh, let's say 16 zebras that have spots instead of stripes. Which is the homozygous recessive condition. Okay, so we go back to our step one. So step one is always we're going to take the number of homozygous recessive individuals in the population and divide by the total number of individuals in the population. All right, so in this case, that's 16 divided by 100. So this here, this is going to be our Q squared. Okay, so we take 16, which is the number of homozygous recessive individuals, and then we're going to divide by 100. Our calendar. Okay, so we take 16 and we divide it by 100, which gives us this number here, 0.16. Okay, now careful, a lot of students stop here and assume that's a value of Q. Remember, that's a value of Q squared. So to find the value of just Q, we take the square root of 0.16. Okay, so remember the square root button, that's the one that looks like a check sign with a line above it. So we hit our square root button, and then that's going to be our value of Q. So we're going out to 2 decimal places, so it's going to be 0.40. So 0.40, so we know that Q equals 0.40, and we know that P plus Q equals 1. So to find the value of P, we just invert it. So 1 minus the value of Q, which is 0.40, which is going to equal 0.60. Okay, so now we know that P equals 0.60, and we know that Q equals 0.40. Okay. All right, so that's essentially the end of the hardest part of it. So we now know the value of P, we know the value of Q. So next step, we're going to plug in the now known values of P and Q into the Hardy-Weinberg equation. Okay, so our equation itself, we've got P squared plus 2PQ plus Q squared equals 1. Okay. All right, so remember, plugging it in just means we're taking the value of P. Wherever we're seeing a P, we're plugging it in. So our value of P is 0.60. So we're multiplying that by itself. 0.0 times 0.60. And we're carrying it down, 2PQ. So 2 multiplied by 0.60 multiplied by 0.40. And then last part of our equation is the Q squared. So our value of Q, which is 0.40 multiplied by 0.40, is going to equal 1. Okay, so then that's, once we have it all set up like that, once we plugged in our values of P and Q into the Hardy-Weinberg, we just do our math. So 0.60 multiplied by 0.60, that's the P squared portion, which represents the homozygous dominant genotype. So that's 0.36. So 0.36, and we're talking zebras, so let's use these. So 0.36, that's the frequency of the homozygous dominant genotype for zebras. So if we want to turn that into a percentage, we move our decimal 2 places. 36% of the zebras in this population are homozygous dominant. All right, and then the middle part of our equation, the 2PQ. So we take 2 multiplied on our calculator. We take 2 multiplied by the value of P, which is 0.60, multiplied by the value of Q, which is 0.40, which is 0.48. So that number represents the frequency of the heterozygous genotype. So in this case, one big Z, one little z. We want to change that into a percentage. We move our decimal 2 places. 48% of our zebra population is heterozygous at the locus that determines stripes versus spots for zebras. Okay, last part of our equation, the Q squared portion. So it's a value of Q multiplied by the value of Q. So 0.4 multiplied by 0.4 gives us 0.16. So that number there, that's the frequency of the homozygous recessive genotype. So little z, little z. Okay, we want to turn that into that frequency into a percentage. We just move our decimal 2 places. 0.16 becomes 16%. So 16% of our zebra population is homozygous recessive at the locus that determines stripes versus spots. Okay, and then we just want to check to make sure we get back to 1. So we take 0.36 plus 0.48 plus 0.16 gets us back to 1. Good news. Okay, and then percentages, 36% plus 48% plus 16% gets us back to 100%. So we know we did it right. So at least that's how I learned to do it, guys. And that's, to me, like, as long as I follow those steps, I never go wrong. Like, to me, my advice is always, always, always follow those steps. And there's really no way to go wrong as long as you're following those steps consistently. Is that helpful?
Definitely.
Yeah.
Okay. Okay, good. So yeah, for those of you in my lecture class, that's also your assignment for this week. You're checking assignment number 2. So if you follow those steps on checking assignment number 2, you're going to get all of those 100% correct. My only warning is just don't forget any steps. You know, just be careful with your rounding and don't flip-flop the values of P and Q. How you guys doing? Do you want another example? Or are you feeling okay?
I definitely think that second one helped a lot.
Okay, good. Does anybody want a third example? I'm more than happy to give a third example if anybody wants.
Just really quick, one question. You said we round the second decimal?
Yeah, so if you're rounding to 2 decimals, all you have to do is look at that third number. So I don't know, let's say we're doing, you know, let's say it was the first one, like we did 20 divided by 1,000. And then we took, or no, it was 20 divided by 1,000, like we did in the first one. And then to find the value of just Q, we had to take the square root. Oh, what did I do here? Okay, well, anyways, like in order to round to 2 decimals, all you have to do is look at that third number. So third number out from the decimal. If it's 4 and below, you leave it the same. So since it's a 1, you'd leave it at 0.14. Like say, for example, if that number was a 7, instead of a 1, it was a 7, you would round up. So 0.14 would become 0.15.
I see. Thank you.
Sure, absolutely. Does anybody out there want a third example? Everybody's good? All right, does anybody have any questions? So you are now officially 6 days out from the due date for lab number 1 through 3. So most of you have been plugging along and I'm grading stuff as it comes in. You know, with the exception of the last couple of days, I apologize about that. My laptop was in the shop, but You know, I'm going to try to grade stuff within a day of you submitting it. So, you know, as you're noticing, the multiple choice questions get graded right away, and then I go through and manually grade the other assignments, the short answer. So, you know, don't be concerned if, you know, if you submit something and you have 4 out of 12, just know that it's just a multiple choice that's graded right away, and then I go through and grade the short answer manually. So if you've got a lot left to do, my advice is really start plugging away at it, because you're now down to 6 days left before the due date. You guys have any questions? You guys are good? Okay, I mean, that's all I've got for you guys today, unless if you guys want me to put you guys into breakout rooms, I can. Is there anybody out there that wants to go into a breakout room with your classmates?
I think we're pretty good for right now.
Okay, awesome. Okay, so I mean, obviously, just if you guys need anything, guys, I mean, especially now that my computer's all working again and my internet's working. I mean, if you guys want to do more Zooms, I mean, I offer Zoom office hours. So, I mean, if you get stuck and if you find this helpful, I'm happy to Zoom with you. Or if you want to, you know, send me your availability, I can try to see if that matches up with any of your classmates. But I'm happy to Zoom with you guys if you need more help other than just, you know, this Thursday Zoom. But definitely let me know. Send me an email, shoot me a, you know, text me on Remind, shoot me an email, so, you know, something like that. Communicate with me in some way so I know that interests you. Okay?
Thank you very much.
Yeah, you're very welcome, guys. You guys have a fantastic weekend, and I will see you guys next Thursday at 3.
Have a great weekend.
Yeah, you're very welcome. You guys have a great weekend.
Take care.
You too.